Before working as a writer at the Fred Hutch Cancer Center, John Higgins had a decades-long career working as a reporter at the Beacon Journal in Akron, Ohio, and the Seattle Times, where he covered government, housing, and education. He also received a Knight Science and Journalism Fellowship at MIT in 2012. John says all that experience taught him that people trust data, but they remember stories. So he applies that idea to help Fred Hutch tell our often data-heavy stories. Welcome to the studio. Before we get into the frog story that you've recently written, tell me a little bit about how a research center divides its researchers up into different groups and why it does that.
So we have a lot of people working on a lot of different problems here that kind of go from basic idea about how life works would be one division. And the next division might say, okay, can we use that somehow? Can we make that into something that could be useful in the clinic? And they'll work on things that we sort of call preclinical. We're going to try it out in mice. We're going to try it out. And then and our research divisions kind of work all the way up to bringing something right to the bedside, a new drug, a new therapy, a new treatment that helps people.
So sort of like a big puzzle with each division doing a different piece of the whole thing that adds up to research.
That's right.
So in this instance, you're talking a little bit about basic science division.
So the basic sciences is poorly named because basic implies simple, and it's anything but. So what they're trying to really figure out is the foundational structures of how life works. And once you figure out how life works, then you can start to think about, well, what happens when it doesn't work? And eventually, what can we do about it?
Scientists study a lot here. And sometimes it's not obvious to me why. Like, why are we studying yeast? Why do we do so much work with mice? And in this instance, why are we working with frogs?
Imagine you had a $100,000 Maserati sports car and you had no idea how it works. Do you want to start monkeying around with the Maserati, hoping for the best? Or maybe you start with a Schwinn bike and figure out how that works.
Okay.
And work your way up. And that's what we do with what we call model organisms. So yeast is a model organism. It's a unicellular fungi. It shares a remarkable amount of DNA with us, even though on the evolutionary tree of life, our last common ancestor was 1.3 billion years ago. So that's yeast on one end. Mice are mammals like us. They're maybe 90 million years ago is when we had our last common ancestor. Frogs kind of fit in the middle. They're around 350 million years ago. We shared a common ancestor. But we share so much basic biology with all of these model organisms that we can study kind of the Schwinn bike. We can figure out some basic things work, not simple, complex, but basic foundational things, how they work in these model organisms, and that helps us get a better picture of how they work in us.
So why frogs in this instance?
We have a new era of frog science because we hired Dr. Yasuhiro Aramura, and he is asking some new questions that frogs are best suited for.
What's the main thing he's trying to figure out?
So what he wants from the frogs is not so much the frogs themselves as the eggs they lay. What he wants is the molecular building blocks the eggs use to make chromosomes. And these frog-made chromosomes provide him with the molecular materials he needs to get at some of the things he wants to study, which are these protein complexes that interact with DNA to form something called chromatin. So if you stretched out human DNA, it would be 5-6 feet long, right? Yeah. And somehow all of that gets compacted down to fit inside the nucleus of a cell. And it does that with these kind of packaging proteins. The funny thing is, when a cell is just functioning, the chromatin is in a certain kind of a shape, which is kind of loose. So the different parts of it can be read and can be turned into the different proteins that do all the cell's work. And then when it gets ready to divide, this chromatin gets packed into the chromosomes, and then the cell divides and each daughter cell gets an exact copy. So the proteins that helped it in one phase of the cell's life change function. They do something different when these cells divide. So it's very difficult to capture this at these different stages. So what Dr. Aramura wants to do is get a 3D picture of what these protein complexes, their shape. Because once you understand the structure, you can understand something about how they work. That presents a problem, though. The things he's studying that are made by the frogs, the frog-made chromatin, they make a sample that's so sparse and spread out, few and far between, that you can't get enough of it in one place. He brought with him some techniques to do that involve tethering the molecular complexes he wants to study under the microscope. He tethers those to this tiny, tiny bead, a nano bead. One of these things is about, what did I figure out, the 2,000th of a width of a human hair. And a little chain of proteins is going to connect the thing he wants to study, like a tether, to one of these beads. And then you magnetize the beads, and then you can draw in your sample into one place in a grid. Once you get your sample together, you're going to shoot a bunch of electron beams through this, and then it's taking two-dimensional pictures, millions of these. And then some computer programs kind of average out these, and then you get a 3D image based on all these 2D pictures. If you have enough sample. And that's been the roadblock up until now, is having a way to get all this small stuff in one place.
Yeah.
And so that's what's really exciting about his work, because you can do more than chromatin. Any small thing where you don't have enough of it, you need to get it in one place. Yeah. You can use these techniques. So there's a lot of people here at Fred Hutch who are very interested in Dr. Aramor's work, because they have some small stuff they'd like to look at too. And maybe some of these techniques will be useful for them.
So question about the frogs and, and the poor frogs in their life. Everybody, thinks about their high school biology class where some of us had to dissect frogs. How is the quality of life for these frogs?
These frogs live the life of queens. Dr. Aramor spent months preparing the frog room, getting these tanks to have the perfect temperature, perfect conditions. And these frogs will be called into duty maybe once every 4 months.
What does duty look like? You
put them in a bucket, you inject them with a little bit of this hormone that instigates the egg laying, leave it overnight, come back the next morning. There's a little puddle of eggs there in the bottom. Frog goes back to her home.
So we're just talking about harvesting eggs.
We're just getting the eggs, and frog goes back to live a happy frog life, maybe 10 years or more.
Speaking about a happy frog life, tell me about Fluffy.
So Fluffy, I had to do a little research for this story. This is why it took a little while to put together.
Yeah.
I wanted to find out, okay, we had frogs here once. What happened to them? You have to read the story, I think, to find out more about Fluffy.
So talking about reading stories, if someone was interested in the coverage that you're doing, how would they follow your work?
Well, I am one of 4 news writers for Fred Hutch News. Fred Hutch News is a feature of our public-facing website, which is fredhutch.org. And all my stories are there.